In 1985, a young Swedish geneticist named Svante Pääbo sat in a lab in Uppsala, staring at a sequence of letters on a screen. He had just done something no one had managed before, extracted what appeared to be DNA from a 2,400-year-old Egyptian mummy, a child wrapped in linen and resin. He published the results in Nature. The scientific world took notice, and for a brief, electric moment, it seemed like the genetic secrets of the pharaohs were about to spill open. They were not. Pääbo would later discover that those sequences were contaminated, possibly with his own DNA. And for the next 3 decades, that failure would hang over every attempt to read the genetic code locked inside Egypt's dead. The mummies, it seemed, had one last curse to offer. Not supernatural, chemical. The very substances ancient embalmers used to preserve the body were destroying the one thing modern science needed most. I think this might be the most frustrating near miss in the history of genetics. 40 years of trying, 40 years of Egypt's climate, its politics, and its chemistry conspiring against the question everyone wanted answered. Who were the pharaohs, really, and what did their blood tell us that their monuments could not? Here is what we knew, or thought we knew. For over a century, the family trees of Egypt's rulers were built the way you would assemble a jigsaw puzzle with half the pieces missing. Tomb inscriptions, coffin labels, the positions of mummies stacked in royal caches, artistic depictions carved into temple walls. A pharaoh calls another pharaoh father in an inscription, and that becomes the working theory. But in ancient Egyptian, the word for father could also mean grandfather or simply ancestor. The ambiguity was enormous. Take Tutankhamun. Discovered in 1922 by Howard Carter, his tomb was the most intact royal burial ever found, over 5,000 artifacts crammed into 4 small rooms. His golden death mask became the most recognized face in archaeology. But whose son was he? Inscriptions near the city of Amarna called him a beloved child of the king, alongside Tutankhamun, who was known to be the daughter of Akhenaten. Some scholars read this as proof that Akhenaten was his father. Others pointed out that the generally accepted chronology placed Amenhotep III's death a decade before Tutankhamun was born, and yet Tutankhamun himself referred to Amenhotep III as his father. A third camp argued for the mysterious Smenkhkare. The identities of at least 4 major royal mummies were simply unknown. They sat in museum cases labeled by tomb number, KV55, KV35, younger lady, KV35, elder lady, names lost, relationships guessed, and behind all the genealogy arguments lurked a much larger, much more politically explosive question. What was the ancestral origin of the ancient Egyptians themselves? Were they indigenous North Africans? Were they migrants from the Near East? Were they, as some groups passionately insisted, Black African? The debates had raged since the 19th century, fueled by colonial-era racial theories, nationalist politics, and the inconvenient fact that mummies do not come labeled with population genetics data. Or at least, they did not used to. Actually, wait, let me back up, because there is something important about why DNA was so hard to get from these mummies. It was not just the heat, though Egyptian tombs can be brutal. It was not just the humidity, which accelerates molecular breakdown. It was the embalming itself. Natron, the salt mixture used to desiccate the body, contained sodium carbonate, which actively degrades DNA strands. Then there was the resin, poured into body cavities, sometimes into the skull after the brain was removed through the nose. Every step of mummification was, from a molecular perspective, a step toward destroying the genetic record. And on top of all that, these mummies had been handled by dozens of people over more than a century. Howard Carter and his team were not wearing gloves. Every touch left modern DNA behind, and modern human DNA is almost chemically identical to ancient human DNA. Telling them apart under older laboratory methods was nearly impossible. So when Zahi Hawass, then Egypt's Secretary General of Antiquities, announced in 2010 that his team had successfully extracted and analyzed DNA from 11 royal mummies, including Tutankhamun, the reaction from the scientific community was split cleanly down the middle. Half were thrilled, half were suspicious. The study, published in the Journal of the American Medical Association, was enormous in scope. From September 2007 to October 2009, the team ran genetic analyses in 2 separate labs, one in the basement of the Egyptian Museum in Cairo, the other at Cairo University's medical school. 2 outside experts, Karsten Pusch from the University of Tübingen and Albert Zink from the Institute for Mummies and the Iceman in Italy, oversaw the work. They amplified 16 short tandem repeats and 8 polymorphic microsatellites, the kind of genetic fingerprinting used in criminal investigations and paternity tests. The results were staggering. For the first time, anonymous mummies received identities. The elder lady in KV35 was confirmed as Queen Tiye, Tutankhamun's grandmother. The male mummy in the enigmatic tomb KV55 was identified as Tutankhamun's father, almost certainly Akhenaten, and the younger lady in KV35, she was identified as Tutankhamun's mother. Here is where the story gets genuinely strange. The DNA showed that the KV55 male and the younger lady were not just married, they were full siblings, brother and sister. Both children of Amenhotep III and Queen Tiye. Tutankhamun was the product of incest, and not the casual one-generation kind. The allele sharing between his parents, 11 out of 16 markers matching, indicated that consanguinity ran back through multiple previous generations. The royal family had been marrying within itself for so long that the genetic consequences were cascading. And you could see those consequences written on Tutankhamun's bones. CT scans revealed a clubbed left foot, a missing bone in his second toe, necrosis, the literal death of bone tissue, in his left foot. The boy king could not walk unaided. More than 100 walking canes were found in his tomb, not ceremonial items, but tools for daily mobility. His tomb also contained what researchers described as an afterlife pharmacy, collections of medicinal seeds, fruits, and leaves. And the DNA revealed something else, fragments of the malaria parasite Plasmodium falciparum, multiple strains of it, making Tutankhamun's mummy the oldest confirmed genetic evidence of malaria in human history. And then there were the 2 tiny mummies. Found in Tutankhamun's burial chamber, sealed in miniature coffins, were 2 premature female fetuses, likely his stillborn daughters. The DNA results were only partial, too degraded for a definitive match, but the team believed they were likely his offspring with his half-sister wife, Ankhesenamun. If that is correct, then the inbreeding had reached a point where viable children could no longer be produced. A dynasty literally breeding itself into extinction. If you have been watching this channel for a while, you know that I find it endlessly interesting when physical evidence and written records finally collide. This was one of those collisions. The art showed a god king riding chariots. The DNA showed a frail teenager who needed a cane to cross a room. And the tiny coffins in his tomb suggested a royal line that had reached a genetic dead end. But not everyone was convinced. Eske Willerslev, director of the Center for Geogenetics in Copenhagen, and biologist Eline Lorenzen, published a formal response in JAMA questioning the reliability of the entire dataset. Their core objection was methodological. The Hawass team had used PCR amplification, an older technique that copies small fragments of DNA but cannot easily distinguish genuine ancient sequences from modern contamination. By 2010, the gold standard in ancient DNA research was already shifting to next-generation sequencing, which reads entire stretches of DNA and can identify telltale damage patterns that are unique to ancient molecules. Without that authentication, Willerslev argued, the results could not be fully trusted. I spent a long time trying to find a good counterargument to this criticism, and honestly, the methodological concern is legitimate. PCR-based studies of mummies had failed before. The field had been burned. But here is the thing that complicates the skepticism. The family tree that emerged from the data was internally consistent across 5 generations. The alleles segregated the way you would expect in a genuine pedigree. Random contamination from lab workers or excavators would be extraordinarily unlikely to produce a coherent multi-generational family line by accident, and the disease findings matched the physical evidence in the tomb perfectly. That does not prove the DNA was authentic, but it makes contamination a much harder explanation to sustain. 2 years later, the DNA trail extended to the 20th dynasty. A 2012 study published in the British Medical Journal examined 2 mummies, Ramesses III and a body known as Unknown Man E, long suspected of being his son Pentauer, one of the conspirators in the harem assassination plot described in the Judicial Papyrus of Turin. CT scans revealed what the ancient embalmers had tried to hide, a deep, wide cut across Ramesses III's throat, almost certainly made by a sharp blade. A Horus Eye amulet had been placed inside the wound, a magical act of healing performed on a man who was already dead. Thick linen and bitumen had been layered over the neck to conceal the injury. The genetic analysis confirmed that both mummies shared identical Y-chromosome haplotypes and matching autosomal markers, a father-son relationship. The murder described in a 3,000-year-old papyrus had left its signature in the DNA. Now, I need to be transparent about a genuine controversy here. The study used Witt-Athey's haplogroup predictor to classify the Y chromosome data and assigned the haplogroup E1b1a, which is most common in sub-Saharan West and Central Africa. But several independent analysts later ran the same STR markers through different prediction tools and got a different result, E1b1b, specifically the V22 subclade, which is the most common variety of E1b1b in Egypt today. Some of the original STR values were statistically unusual in ways that suggest possible degradation or error. Without direct SNP testing, which was not performed, the haplogroup assignment remains genuinely uncertain. I rewrote this section 3 times because the evidence genuinely pulls in 2 directions, and I think it is important not to oversimplify what is actually a complicated and politically sensitive question. This matters because every piece of genetic data from ancient Egypt gets pulled into a much larger argument about race, identity, and who owns the pharaohs. Far-right groups seized on earlier claims about the 18th dynasty to declare the pharaohs were white Europeans. Afrocentric scholars pointed to the Ramesses data as evidence of Black African rulership. Both sides were working from incomplete data and enormous political stakes. The actual science says something less dramatic but more interesting. Ancient Egypt sat at a genetic crossroads, and its population was never one thing. The breakthrough that finally silenced the skeptics about DNA preservation came in 2017, when Johannes Krauss at the Max Planck Institute led a team that did what Pabo had failed to do 3 decades earlier. They used next-generation sequencing, the method Willerslev had demanded, to extract DNA from 151 mummy heads from Abusir el-Melek, a burial site about 100 kilometers south of Cairo devoted to Osiris, god of the dead. The technique works differently from PCR. Instead of targeting specific fragments and copying them, next-generation sequencing reads every stretch of DNA present in a sample. This matters because ancient DNA carries a specific kind of damage, particular patterns of chemical decay at the ends of the molecules, that modern contaminating DNA does not. When you can see the full read, you can authenticate what is real and what is not. The mummy heads had been excavated in the early 20th century and transported to collections at the University of Tübingen and Berlin's Museum of Prehistory. while the soft tissue yielded almost nothing. Bones and teeth were, as Hannes Schroeder of the University of Copenhagen described it, full of genetic material. 90 of those mummies yielded usable mitochondrial DNA. 3 yielded genome-wide data. The samples spanned 1,300 years, from the New Kingdom through the Roman period, covering multiple foreign conquests by Assyrians, Persians, Greeks, and Romans. The results overturned a major assumption. Despite centuries of conquest, the genetic makeup of these ancient Egyptians remained remarkably stable. The conquests left surprisingly little genetic imprint. Ancient Egyptians clustered most closely with populations from the ancient Near East and the Levant. But here is what caught everyone off guard. Modern Egyptians carry 15 to 20% more sub-Saharan African ancestry than their ancient counterparts did. That shift happened not during the great conquests of antiquity, but after the Roman period, possibly driven by the expansion of trans-Saharan trade routes, the slave trade along the Nile, or the spread of Islam, intensifying contact between northern and sub-Saharan Africa. And yes, I know that sounds dramatic, but the numbers back it up. Then, in July 2025, the final barrier fell. Researchers from the Francis Crick Institute and Liverpool John Moores University published the first complete genome from ancient Egypt in Nature. Not a partial sequence, not targeted enrichment of a few 100,000 genetic markers. A full genome from a man who lived between 4,800 and 4,500 years ago, during the age of the pyramids. He was not a pharaoh. He was probably a potter. His skeleton showed decades of hard labor, expanded seat bones from squatting, arthritis in his vertebrae, heavily developed arm muscles. He was between 44 and 64 when he died, an unusually old age for the period. And he was not mummified. He was buried in a sealed ceramic pot inside a rock-cut tomb at Nuweirat, 265 kilometers south of Cairo. That pot, sealed inside that cliff, created a stable microenvironment that preserved his DNA where mummification would have destroyed it. The irony is almost too perfect. The technique the ancient Egyptians invented to preserve their dead was the very thing preventing us from reading their genetic code. The man who skipped that process gave us the clearest signal. His genome told a specific story. Roughly 80% of his ancestry traced to North African Neolithic populations, the indigenous farming communities that had lived in the region for thousands of years. But approximately 20% came from the Eastern Fertile Crescent, including Mesopotamia, modern-day Iraq, and Iran. This was the first genetic confirmation of what archaeologists had long suspected from pottery styles, writing systems, and trade goods, that people, not just ideas, moved between Mesopotamia and Egypt during the formative centuries of Egyptian civilization. If you want to follow what happens next, the channel is covering these developments as they unfold, and there is a lot more coming on ancient migration patterns that connect directly to what this genome revealed. Subscribe so you do not miss it. So, what does all of this add up to? 3 things. First, the royal bloodline was far more closed than anyone fully appreciated before DNA. The 18th dynasty pharaohs were not just occasionally intermarrying, they were trapped in a genetic loop that produced accumulating deformities across generations, and those deformities shaped real political events. A boy king who cannot walk is a boy king who cannot lead armies. The end of the 18th dynasty was not just a political collapse. It was, at least in part, a biological one. Second, the general population was far more genetically stable than the political history would suggest. Conquerors came and went, the genetic signature barely moved. This challenges the common assumption that military conquest equals population replacement. In Egypt, it did not for over 1,000 years. Third, and this is where it gets uncomfortable for anyone looking for simple answers, the ancient Egyptians were genetically mixed from the very beginning. North African, Near Eastern, Mesopotamian. The Nuweirat genome shows that blending was already present at the dawn of the Pyramid Age. The question was, never were the ancient Egyptians African or Middle Eastern. They were both from the start. But we are still working from fragments. One complete genome, 90 partial mitochondrial sequences from a single site. A handful of royal mummies tested with methods that half the field still questions. Right now, in Cairo, Yahia Gad supervises an ancient DNA lab at the National Museum of Egyptian Civilization as part of the Egyptian Genome Project, an effort to sequence the genomes of 200 mummies and 100,000 living Egyptians. The technology that took 40 years to crack a single genome is about to be deployed at industrial scale. The Nuwayrat man's bones are back in Liverpool, in the same museum that survived the Blitz. His teeth, which gave up the DNA, were excavated in 1902 under British colonial rule. His genome was decoded in 2025 by an international team. And somewhere in Cairo, right now, researchers are drilling into the teeth of pharaohs, looking for the same fragile molecules. The dead are finally talking. And we are only just learning how to listen. What would change about our understanding of ancient Egypt if Gad's team sequences even 50 royal genomes, and the family trees we have been building from inscriptions for a century turn out to be wrong?